Antonella if she's able to hear us.
Yes.
Yeah, I can hear you.
I'm a parent of a child with a GRIN disorder, and I'm conscious that a lot of us are affected by epilepsies. And I wondered, how can you tell whether an individual child is likely to be affected by their gut biota? Do you just need to test and try and change?
So there are a lot. Yeah, exactly. As you have seen in the presentation, there are some some easy screening tools, such as that of the poo. So you're through this test, you're understanding if you have problems in your gut locally. You have an idea if your feces are normal, or if there are problems, which means constipations or diarrhea. When there are these things, it's reliable that also the bacteria species in the gut are modified. So there are some facilities which are offering tests. So you can do the collection at home and then send to them, and they are giving you a report. But what is important about this report is that then you should give it to a dietitian or a nutritionist, which is really able to read these results and so suggest you what diet you can undertake to try to elevate some bacteria species which are beneficial and reduce the are not beneficial, and also to try to add biotics in the diet. Also, at the moment, we do not have the right biotic for each disorder. So there is not a specific biotic which is better than the other, but the response it's really individual. So we should go for a trial and error basis, see if that one patient is responding well that biotic and the diet, seeing changes in the poo, for example, or in case if this is not good, changes. But what is important is yes, the realities which are giving you reports, but you should dietitian to look at it to make great changes to the gut microbiota.
Thank you.
Sorry, we're going to prioritize the questions for Giske. So thank you.
Somebody, is there any question, please? No. Sorry.
Thank you guys all for your talk. So with regard to what drugs, you were saying that there could be some overlap between potential compounds that could work across the different genes. Do you guys have any that you have already targeted or that you're thinking about targeting?
First of all, I think that for kinase disorder, many of the drugs that actually target the NMDA receptors by reducing its function if kinase is too active, or by enhancing its function if kinase is not active enough. I think in that respect, we can definitely also profit from what's known already from the GRIN field, but I think also vice versa, sometimes targeting not so much so if you have a mutated GRIN protein, so one of the subunits of the NMDA receptor is not functioning, I'm not sure that the drugs could definitely normalize that. And I just think that sometimes it might be useful to just go to the direct first consequence intracellularly that might actually be enhancing then functions in a maybe more efficient way than by targeting the NMDA receptor, just circumvent the part that's not functioning. And yes, and then when it comes to which type of drugs, well, we're also working on some kinase specific drugs, but unfortunately we are not there yet, but we're definitely working on it.
Thank you.
Are there more CAMK related questions?
You want to collaborate with Giske because she needs to run. Just if it's okay, we will share your contact. Okay?
Yeah, I would like to collaborate. Can you see CAMK2 as a biomarker for activation or for synaptic plasticity in any way?
Yes, so we know that indeed the moment that you induce genetic plasticity, you will see enhanced autophosphorylation of CHIM kinase. So that is one of the readouts. The other thing what you see the moment that indeed genetic plasticity is being induced, that sometimes you get this structural plasticity, that the synapse really physically grows. Also that's CHIM kinase dependent. So there are some biomarkers that directly can indicate that CHIM kinase is involved.
Is there an easy way to detect it?
The autophosphorylated form of CHIM kinase, yes, we've got a beautiful antibody that detects it. So you can beautifully show it already on Western blot. So if you induce LTP in your model and you just harvest the tissue within a certain time frame and then put it on Western blot, you should see indeed CHIM kinase is being activated. So one thing that I would like to really motivate, especially GRIN and GRIA researchers, start to look in your mice also at CHIM kinase, what happens, stability and autophosphorylation. I think those two are very important readouts.
Does the autophosphorylation translate to phosphorylation of the other AMPA and GRIN?
Yes, so if alpha-CHIM kinase becomes activated, and that's the slide that I skipped, then we see that indeed alpha-CHIM kinase can also phosphorylate beta-CHIM kinase, but it could also potentially phosphorylate delta and gamma, but they are very low expressed in the brain, so more difficult to detect.
But it would be a
But
good biomarker for phosphorylation of the other receptors. Okay, cool.
I just have a quick question. I have the mic here. So I just want to ask you, could it be potentially reduced number of CAMK2 proteins if you have a hypofunction of either one of the receptors? Do we know?
I don't know if Maria is still here, because there was a poster indeed for the GRIA3 knockdown from the group of Fabrizio Gordoni, I think. Okay, because she on her poster actually has a beautiful experiment where she knocked down of the GRIA3 subunit of the AMPA receptor, and she showed reduction of CHIM kinase levels, which was highly surprising for me, but I think that's very, very interesting. Okay. And I'm trying to motivate other people also to look into that.
You have Maria's contact?
Yes, I do.
Okay, good.
Okay, I think we're going to let you run to the airport. Thank you so much.
And thank you so much for having me. And have an honor for me.
And you for us to learn from nature to collaborate as the nature does in that system. Good. Do we have further questions? I think
I have a question on the iPSCs, which also would interest Geeske, but she's gone. You gave like a Star Wars neurostorm of data that was really impressive, which made me think that if you are a parent of a child with GRIN, that it is really necessary to realize that these are actually very embryonic, very immature neurons in a dish, which is not the same as behavior in real life. But seeing your data, I saw the control and then all the different drugs, and what I actually missed was like the negative controls. Like, did you test any drugs in which you know that they could potentially influence the phenotype in a negative way, just to see that not everything you throw in a dish is positive, but just to have a more balanced readout?
So we have first, treatments with just the MSO or the solvents, which don't affect. With the other drugs, we didn't do drugs that are specifically, we thought would not affect. There were some drugs that had little effect, and there were drugs that had more effect. But the negative controls were just the solvents themselves.
So not like in a gain of function mutation that you use something that increases the function, or in a loss of function mutation, something that even more blocks the NMDA channel.
Right. I have also to say about the maturity and so forth. So when the neurons are approximately 10 weeks, they are, still considered prenatal and so forth. But in the dish, of course, there is, no homeostasis mechanism. So the neurons do much. Of course, for a human neuron, it takes 9 months, right, to reach. So they are more mature actually than what they are in months. And we do have some results not from this disorder, but from another disorder where we saw a good impact on the cells, and then we used it on a human patient and he's 50-something years old and had incredible effect. And also here we see also with the rats. So we do think that overall, the evidence do suggest that there will be a good effect also on the patients.
for some of these drugs.
Yeah, for the drugs that are more effective. Yeah.
I would have a question on a little bit in general. The ones that do all these clamp assays, it's beautiful and nice. Do you always control for surface expression of the protein, of the variants? In general? Possibly the one that does will say yes.
No.
Of course.
No, well, I mean, and I guess I should, because it's clearly going to matter how many of the receptors make it to the surface. But while not accounting for it, you saw how clear the gain of function, loss of function thing was. So clearly that massively dominates the symptomology of the patients. So even if some of these gain of function variants only have 10% surface expression, if I can see their gain of function in a HEK cell, seemingly that overrides anything else. And I mean, to be lazy and frank, I don't want to go back and do 51 biochemistry experiments, having just done 51 electrophysiology experiments. So there are a whole bunch of properties that could be investigated. And this is one that we're hoping we don't need to get the right answer for the patient. So
maybe I could just add something to that. So the other thing that we sort of embraced quite a lot in the last 10 years is that the AMPA receptor, with NMD receptors, they don't really have auxiliary proteins, right? There's just no evidence for that, right? But the AMPA receptors really have a lot of auxiliary proteins. And for example, GluA3, GRI3, if you try to express that in cells, it doesn't express very well. But if you co-express it with the TARP, for example, it expresses perfectly fine, right? So, because that's what the TARPs do, right? They bring it to the surface. So, in all the work that we do on heteromeric receptors, so not homomeric, then we don't really see much defects in terms of function. We've measured that. It's buried in our papers and things. I guess the thing that maybe I didn't get over in my talk is that the AMPA receptors' properties are changing dramatically during brain development. And we know they're changing dramatically even when you induce plasticity mechanisms. So when you start to think about targeting the AMPA receptor with drugs, right? It's changing all the time. It's biochemical representation is changing all the time based on circumstances. And given the fact that you've kids with neurodevelopmental disorders, you can imagine that synchronization and that kind of regulation is all messed up. So I think that we need a few years to work that out. But I think it's an exciting time because it allows us to really think about gene therapy, and some people were talking about antisense yesterday as well. So I can see this could be a great model system for that, actually. So.
Okay. Thank you all for the amazing presentations. This is more of a parent type of question, so not so scientific, but goes back to a bit to the previous question, inspired by Dr. Stern's presentation. And I would be wondering, to the extent that there might still be any physicians around here, have any of you already used any of these substances to treat any of your patients? And if so, what sorts of results have you seen? I know maybe this is a bit complicated, so it could be a panel in a future conference, but I think it would be interesting for the parents perhaps to know this. Thank you.
With explanation. So I can maybe, Tali, you want to say a few words about this? Yeah.
Is it a question to the doctors or to Dr. Stern? Because
I'm the one that suggested several of the drugs. And indeed, for CBD, for instance, we do have at least 2 patients that are given it, and those we see an improvement. About minocycline, I was reluctant because I wanted to wait for the animal model results, and I do see that it is valid. And I do know of a child that is treated with minocycline and is improving, but I don't have more information about it.
Yeah. So one of the drugs you saw on your slide was methylphenidate, which was not tested for all the patients, but for some, which had actually quite good effect, and that is Ritalin, like Concerta, the one used for ADHD. And in our patients, we use that quite a lot, and we see actually of all the drugs, you have antipsychotics, antidepressants, ADHD medication, and those are relatively well responding with not the large amount of side effects that we see in the antipsychotic or SSRIs. We do use CBD oil in, or I use CBD oil in several different syndromes, but I'm very cautious about it because it can also induce like a lot of epilepsy, even in people who have very low frequent epilepsy. So all these drugs, they have their own side effects and also their own controls that need to be done. Even L-serine needs blood controls to do it safely.
I think CBD is given for many autistic children, and in Israel we have a lot of children that are using CBD and the safety profile is quite good. I must say that in 5% it might cause a elevation of liver enzymes, only in 5%. So other than that, many physicians do prescribe CBD.
May I ask for questions for the speakers quickly? I would love to take these discussions into the afternoon open session after we hear the last two presentations. Sorry. Yeah, and Ian had a statement. Sorry.
Oh, I had actually, it was a fresh question. It was, I was jumping the queue. So Ladislav, you go.
Wonderful.
May I? Yeah, I have a question concerning the sensitization. I wonder why you don't use nootropic drugs like aniracetam, piracetam that slow down the desensitization as a way how you can deal with the mutation-induced defect.
Yeah, aniracetam is an interesting drug. It was made as part of a neurotropics program in the 90s. When you actually look at what it does, I mean, at 5 millimolar, yeah, it blocks desensitization, but it gets chewed up by the liver when you swallow it. And I have my extreme doubts that any neurological effect of aniracetam is due to an effect on an AMP receptor. I just really don't think it's such low potency. Honestly, I don't think, and piracetam as well, I really don't think they act through AMP receptors, but I mean, I'm prepared to be debated on that, but it seems quite unlikely. So I would say, and that is partly one of the reasons it's probably made it through trials and been safe, is that it doesn't fry your brain, which is always the danger when you boost AMP receptors, because so much of the signaling is AMP receptor based.
But it sometimes irritates people when they use it.
It's what irritates?
Irritates, they are jumpy.
Oh, right, okay. Yeah, I'm not saying it does nothing. I'm just saying whatever it does, I don't believe is through an AMP receptor. That's, yeah. Fine.
So Derek, the mouse is pretty intelligent at stealing cheese from your kitchen, and the elephant is pretty intelligent in finding water. So is there, in terms of regulation of AMP receptors, anything that would suggest that the associated proteins, there are some human-specific ones that we still got to try and identify, or particular assemblies that are favored that then allow for additional modulation of AMP receptors?
Yeah, so that's a great question, right? So it's true, the mouse, and I mean, that's why we use mouse models. They've got the same AMP receptors, the same subunits. We don't know if the processes of post-transcriptional modification, et cetera, are the same. But the other thing that's changing is our view of the synapse. Its morphology is changing. It looks like it's much, much more sophisticated than we thought. How the AMP receptors and the compositions are put together. There's different kinds of neurons, of course, in the human brain. There's much greater kind of connectivity. So I was being a little bit unfair in presenting that, but it seemed like a nice way to introduce the idea of, if you just look at the genes, you miss all the important things. The reason that we are what we are, rather than the simple vertebrates that we are, is because of this post-transcriptional modification, right? But once you get into the humans, and we're unable to get human tissue to really look at that. But I have no doubt there are other biological inventions there that kind of address exactly but what they are, we don't know yet. Okay.
Thank you all for the talk. My question is about Ian's talk regarding the 508 variant, where you don't see any gain of function or loss of function. Have you tried testing any agonist affinity, maybe?
Yeah, that's a key experiment, and it's one that on these patches is they last okay, but they don't last ages. And so by the time you jump to like 6 or 7 or 8 different concentrations on there, the receptors you're looking at the end of your recording are potentially, it's not so easy. So, but in terms of that particular variant was looked at by CFIRB in oocytes, and they showed a slight increase in affinity. It wasn't huge, but it was potentially enough to explain. But that one and just a couple of them have been, we're pretty confident we looked at them as well as we can, and we can't see the change, at least in terms of the kinetics, because the kinetics is a much more important property in terms of the receptors at the synapse than their steady state affinity, because there is no steady state of glutamate in these synapses. It kind of, when there's an input, it leaps up to 3 millimolar, and then within 10 milliseconds, it's down kind of in the low micromolar again. So we always feel that our approach is the better approach, but there is a very, very inconvenient truth is that you don't get any different answers from an oocyte that you get from these patches so far. And potentially even in that particular case, you get an answer from an oocyte that we weren't able to get in the HEC. So anyway, I'm setting up a lab looking at oocytes now, and then hopefully I can join in and join in with like that.